Good afternoon and welcome to RSSL's webinar on cleaning validation. This is the final of 3 sessions that we've been running on this topic, and today's webinar will focus on cleaning validation, validating the efficacy of disinfectants. Darlington Nwodo will be delivering the webinar this afternoon and I'll be here to support him. My name is Lisa Hallwood and I'm one of the senior account managers here at RSSL in the commercial team. Darlington is our pharmaceutical microbiology laboratory manager at RSSL and has many years experience, including hands-on experience in working with customers on their cleaning validation projects. For any questions throughout the presentation, please send a short message within the chat box on the right-hand side of your screen. We'll do our best to answer questions at the end of the formal presentation. So now that we've got the admin out of the way, let me give you a little bit of background around RSSL and to better understand why we're positioned to share our knowledge with you today. RSSL was established in 1987 and has been supporting a number of industries, including food, healthcare and pharmaceuticals on a global basis during this period. We currently work for 20 of the top global companies as well as small startups and SMEs. Our MHRA and FDA inspected facility in Reading is where our team are based, and our technical expertise covers microbiology, chemistry, physical sciences and biochemistry. RSSL have been working in the pharmaceutical sector for more than 25 years, and our multidisciplinary teams support our customers from early phase development all the way through to finished product QC analysis. For some clients, we undertake all their product development and testing, and for others, they would potentially use us for more specific methodology where perhaps they don't have the expertise or equipment internally. Our unique emergency response service ensures priority access to laboratories 24/7 to support projects which are time critical, such as resolving customer complaints or manufacturing issues. In supporting the pharmaceutical industry, RSSL has a breadth of capabilities and a wealth of experience across a number of different areas, from sterols or topicals all the way through to oral dosage forms. We support customers with their own methodology, and we regularly work to pharmacopoeial monographs, the USP and EP being the most commonly used, and others such as the Chinese, Japanese and Brazilian pharmacopoeias being more frequently requested as our customers move into new geographical regions worldwide. RSSL like to partner with our customers, better understanding their needs and refining the project scope to keep things as simple as possible whilst ensuring that the regulatory requirements have been met. This is especially relevant to the approach that we take with our cleaning validation projects. I'm now going to hand over to Darlington, and he's going to present the technical content of this webinar and tell us a bit more about cleaning validation, validating the efficacy of disinfectants.
Thank you very much, Lisa, for that introduction and a bit of background of RSSL. Good afternoon, everyone, and thank you for joining us on this webinar. I'll be covering cleaning validation, microbiological cleaning validation, and validating the efficacy of your disinfectants. Just an overview of what we'll be talking about today. First of all, we'll be looking at the purpose of the validation. Why do you have to validate your disinfection process? Why do you have to validate the efficacy of your disinfectants? We will then move on to how to evaluate your disinfectants if you already have disinfectants in place, how to evaluate the disinfectants in your cleaning process to see if you have the right disinfectants and the right range of disinfectants. Now, we also look at key consideration before embarking on a project. These clean validation could be a costly project, but it doesn't have to be that way if you can avoid the pitfalls. And after that, we will move on to the methods that are out there that can be used to undertake a cleaning validation study. We will look at a flow diagram of how the qualification process should be, stages you should take, and after that we will talk about the practical tests, more of a summary about the practical tests. We won't go into too much detail about the practical tests. And at the end, we'll summarize what we've talked about today in the presentation. So let's get started. Now, purpose of validation. Now, as we all know, disinfection and cleaning regime is a key aspect of our contamination control, and microorganisms, getting rid of microorganisms is quite important because it can have an impact on patient safety. It is important to evaluate the efficacy of a disinfectant to ensure they are fit for purpose. And this can only be achieved by testing them under the conditions they're used on your manufacturing site. So there's a number of tests your disinfectant manufacturer might have performed as part of their bottle claim. It's good to remember that this test does not apply to your cleaning validation because they've not been tested in the conditions they use. So therefore, this does not establish that they are fit for purpose. So you have to perform a cleaning validation to establish if your disinfectants are fit for purpose or not. And more importantly, this is a regulatory expectation. I've noted a few documents that have highlighted the importance of microbiological cleaning validation. You can have a look at these documents. I've mentioned the sections of this document, so have a read. But a summary of what these documents have, the content of this document is really, they all recommend that the efficacy of a disinfectant, your disinfectants should be validated. And you should ensure that residues do not interfere with the recovery of any type of microbial testing you're doing. So in form of environmental monitoring, your contact plates or swabs. Finally, regulators expect to see a documented process. So depending on what you achieve from your cleaning validation, the final phase really is to document what your cleaning and disinfection process is. So let's talk about how you can go about evaluating your disinfectants and the process you have in place. So I think this really starts with a simple question of, do you have the right disinfectants in place? So if you work in a grade A or B clean room, you really should have sterile disinfectants, because it's important that you maintain a high level of contamination control in these grades of a clean room. Do you have the right bactericidal and fungicidal agents? You should have a wide range of disinfectants that are effective against bacteria and fungi. And even more importantly, this I've seen being missed out in the evaluation process, is, do you have sporicides in the house? Good to remember that many disinfectants are not effective against spores. For example, 70% IPA, which is widely used in many sites, is not effective against Bacillus species spores. Now, do you have the correct contact time in your cleaning process? And it's important. A good example is to account for the evaporation of disinfectants like IPA. If you have a contact time of 10 or 15 minutes for IPA in a clean room with a high level of air changes, it really is not fit for purpose. So you need to look at that as well as part of your evaluation phase. Are your disinfectants effective after their hold time? So if look at your hold time of your disinfectant, and if you use spray bottles, if you transfer your disinfectants into spray bottle, you really should look at, are they effective after that transfer and the end of the hold time. Other key consideration is to, before embarking into a cleaning validation project, I've just highlighted a few key considerations which you need to look at, because a lot of times sites embark on cleaning validation without considering this point, and it ends up being a very costly exercise. But if you can avoid the pitfalls, it doesn't have to be expensive or time wasting. So the first key consideration is a request for the available data you have for your existing disinfectants. Now, disinfectant manufacturer, they do perform a number of tests as part of their bottle claim. Request for that data. It might be a standard suspension test or a surface test they've performed to show the efficacy of their disinfectant. Ask them for this data. The second point really is quite important. It might sound very basic, but I have seen this being left out in this process, and it could end up costing a lot of time. So speak to your microbiologist on site. And if you use a contract testing lab or a specialist lab, they might have come across this disinfectant or the organisms you're looking for the disinfectant to be effective against. Talk to microbiologists. Look at this data with them. Other key consideration is the in-use state of your disinfectant. If they're stored in spray bottle, you want to perform your cleaning validation at the end of the whole time, just to cover the worst case scenario. When looking at the surfaces, always remember to use the same surface you have on site, because difference in surfaces can have a huge impact on the results. So endeavor, work with your supplier to get the samples of the same surfaces you have on site. And the conditions. Normally a contamination organism is presented in a certain condition. It might be a clean or dirty condition. So you have to simulate this as part of your cleaning validation. Methods like the BSEN standard add bovine serum albumin just to simulate a clean or dirty condition. So you need to think about this as well. Now, a key aspect as well is to make sure you incorporate your environmental isolates. Ensure you include this in your cleaning validation process, because the real fact is your disinfectants will encounter these isolates. So to establish their fit for purpose, you have to include your environmental isolates as part of your cleaning validation. In use temperature, what temperature can have an effect on the performance of your disinfectant? And if you work on a site, on a manufacturing site that have you manufacture on that temperature that is way outside the ambient temperatures, include that in your process. Also think about the concentrations you use them in. You want to qualify the concentration you use your disinfectants in and include the grade of water you use in making up these concentrations. It is acceptable to use a lower concentration just to cover a worst case scenario, but I mean, for minimum, use the concentration you use on site. Another part of this is to perform it in triplicate as a minimum. Just like any validation, the more data you can gather, especially in microbiology, the more confidence you will have in the performance of your disinfectants. And always follow a risk-based approach when picking out your surfaces. Look at those surfaces that are most critical to your manufacturing process. Make sure you include those surfaces. Always remember that organisms are more likely to settle on horizontal surfaces, and operators are always mainly the key risk around contamination. So if there's a surface that operators normally come in contact with frequently, you want to include that as well as your in your validation process. So those are the key considerations. Now let's move on to the methods. There's a number of methods that are used as part of cleaning validation. The BSEN standards are widely accepted. They're good sets of tests. A quantitative test used to evaluate the bactericidal, fungicidal, or sporicidal activity of chemical disinfectants and antiseptics. These standards are mainly designed for chemical disinfectants and antiseptics. So if your disinfection process is outside of this, you need to look at different sets of tests. It comprises of several phases that evaluates the efficacy of disinfectants in a suspension and non-porous surface tests. So it's a combination of suspension tests and non-porous surface tests. And just to mimic how they're used in the manufacturing sites, these standards incorporate bovine serum albumin to create a clean or dirty condition. The methods can be adopted to simulate in-use conditions by adjusting things like the contact time. So if your cleaning process, you usually have a 5 or 10 minutes contact time, you can use these methods. However, amend the contact time to suit what you do in practice. You can always incorporate your environmental isolates in addition to the standard organisms that form part of these methods. These methods normally have stainless steel as the standard surface, but you can add the other surfaces which needs to be part of your cleaning validation exercise. Just more detail about the methods. Now, for evaluating chemical disinfectants and antiseptics, the phase 1 tests, example of that is a BSEN 1040, a suspension test. It's a basic evaluation test used to evaluate the efficacy against bacteria. BSEN 1275 is also a suspension test it's a basic evaluate. It basically evaluates efficacy against yeast and mold. So for these tests, you have the standard gram positive and 1 gram negative organism, and a yeast and a mold. At this phase 1, this is mainly used by disinfectant manufacturers at their development stage. So this is more of a look-see kind of test. They might have a new formulation. They perform this test to see how effective the disinfectant is, and if they need to reformulate, they'll go back and do so. So I wouldn't expect this to be incorporated in a cleaning validation. However, it's good data you can get from your manufacturer. Now, phase 2, step 1 can be incorporated as part of your pre-validation tests. Example of that would be the BSEN 1275 suspension test, which evaluates efficacy against bacteria. It also adds interfering substance, so you can mimic the condition the organisms will be presented in. So it has a wide range of organisms, of bacteria, and the equivalent of that is the BSEN 1650 suspension test, which evaluates against yeast and mold. A recent update to that method has added the use of spiny spores, and this it makes the test even more, the criteria is more stringent, but it covers the worst case scenario. If your disinfectant is effective against spiny spores, then you can have more confidence in it. Another test which forms part of phase 2 step 1 is the BSEN 13704. This test is designed to evaluate for sporicidal activity of disinfectant. So if you have a sporicide, ideally they will be putting this against this. You'll be testing it against the standard. Like I said, your disinfectant manufacturer might have done this test as part of their bottle claim, and it's important to understand, because this phase of this test is a suspension test. It does not mimic how the disinfectants are used in practice on your site. On site, you normally do your disinfection process on a surface. So a suspension test, it's easier for disinfectants to meet the acceptance criteria for suspension test, because the organisms are presented in a suspension, unlike on a surface. So it doesn't mimic, and it can be used. It cannot be used to establish that your disinfectant are fit for purpose. And it's always good to note that efficacy of undiluted disinfectants can only be tested at 80% concentration or less. This is just because a dilution is always produced by adding inoculum and interfering substance. So you can add more like an uplift when your disinfectants are made at a concentration, but for a ready-to-use disinfectant, you can only test at 80% concentration. These tests can be used in the pre-validation phase, because you can tell if the disinfectants are effective enough to move on to the next phase of the cleaning validation, and it can save you a lot of time. Now, let's move on to the methods that are used for cleaning validation tests, and these are referred to the step 2, the phase 2, step 2, beg your pardon. A good example of this test is the BSEN 13697. This test is for non-porous surface tests, which evaluates bactericidal and fungicidal activity. It has a wide range of standard organisms, from a gram-negative rod to gram-negative to gram-positive organisms as well. It's also incorporated a yeast and a standard mold, Aspergillus brasiliensis. And again, an update to this method, the recent update has added that your disinfectant will need to be challenged against spiny spores. Now, these tests condition, it does mimic condition potential contamination will be presented in a manufacturing site, and different surfaces can be tested as part of your qualification process. So this is actually a good test to use for your cleaning validation. Another test, which is also a phase 2 step 2 test, is the BSEN 16615 for non-porous surfaces as well. This evaluates a bactericidal and yeasticidal activity with the addition of a mechanical action. So this does mimic what happens in a manufacturing site. So in manufacturing site, you might be using making use of wipes as part of your disinfection process, and this test has included it. However, this test has a swab phase of organism recovery, which can present a challenge with reproducibility, and it does have a lot of variation. I know my colleague has covered cleaning validation from an analytical point of view using swabs. But in microbiology, where you can, you want to avoid the use of swabs in your test methods, because I've looked at a lot of swab, and I have performed a few swab recovery studies in the past. So they're not the best methods to use. So that's the huge drawback for this method. Now, so let's flow diagram around how a qualification process should go about. So on the left is a site with an existing cleaning and disinfection process in place, and on the right is a site with no process in place as of yet. So I would recommend to always start off with a risk assessment. You want to assess the risks on your sites, because this risk assessment, it will depend on what you manufacture, what happens on your sites. It include things like the amount of operators on your site, the disinfectants you have, and from that risk assessment, you have defined parameters, because you've identified the risk, then you have parameters, you have acceptance criteria to mitigate the risks. Now, for a site with existing process in place, what you want to do is do what we talked about at the start of this presentation, which is the evaluation of the disinfectants you have in place. So evaluate the disinfectants you have in place. Do you have the right range of disinfectants? Do you have disinfectants that are effective against bacteria, yeast and mold, and bacteria spores as well? Do you have sporicides in place? And if you don't have a process in place, and this is, for example, where this can apply is where you have a new facility. So be part of the design, and at this stage you can look at acquiring a range of disinfectants to meet the criteria you've set. So moving on from this stage, you want to perform a pre-qualification test, a set of pre-qualification tests, and you can apply you can go for an option of applying a little surface test, but you can look at the suspension test we talked about earlier. This can be a good indicator. However, it's good to add that sometimes organisms can perform extremely poor when you get to the surface phase of the test. But in the interest of saving a lot of time and costs, it's important to perform a pre-qualification test and look at the data you've you received from your manufacturer, the disinfectant manufacturer. This will give you an idea of the efficacy of your disinfectant. What are the tests you need to perform? If you have a disinfectant where the bottle claim test is only focused on setting organisms, you might want to look at incorporating your environmental isolates in a suspension just to see if they're effective enough. And after that phase, you want to move into the performing the full cleaning validation. Now, at this stage, I would strongly recommend performing a surface test just to mimic what goes on the manufacturing site. And depending on the results of your cleaning validation, you might need to amend your disinfection process. You might see that the contact time you have in place is not effective enough. You might actually look at sourcing some new disinfectants. And once you have that, once you've done that, you want to document all this process, document the new process and all the changes, and control that change as well. For a facility that's a new facility, depending on your results, you want to look at documenting your cleaning and disinfection process based on the results, based on scientific data. These are the kind of things regulators will want to see on site. So that's just a summary of how the qualification process should occur. Now, just quickly, before embarking on a cleaning validation, I would say this is a quick checklist on a list of things that you must have identified before moving on to the cleaning validation. You must have your contact time in place, and it should reflect your disinfection and cleaning process. The organisms you will be using should cover a wide range of organisms, so it gives you a bit of room for the potential contamination. And make sure you include your environmental isolates, because in real practice, these are the organisms, the disinfectants we encounter on your site. And if you've isolated an organism from a previous contamination issue, you want to include that as well as part of your cleaning validation process. Now, the surfaces, usually for the 13697, you can use a 2 by 2 centimeter coupons of the same surface you have on site. Now, I know sometimes this can be a challenge, but it's very important to get a sample of the same surface. Talk to your supplier, see if they can provide you with the same grade of surface or an exact surface. Now, in regards to the surface conditions, simulated surface conditions, considering what we talked about earlier about how the contamination is presented to the disinfectant. If you work in a clean room environment, you should be looking at a clean condition. So the EN standards, we add a lower level of interfering substance. EN standards uses bovine serum albumin, and you can also go for a worst case scenario by going for a dirty condition. However, bear in mind that the conditions have a huge impact on the results you get. You can test the same disinfectant using but changing the conditions, and you can get a pass for clean condition and a fail when you test again in a dirty condition. So it's just something to bear in mind when you're making these choices. Now, the acceptance criteria. On the right, I've added the BSEN standards the acceptance criteria. There's a 4-log reduction for bacteria and 3-log reduction for bacteria spores and fungi. Now, bear in mind the BSAS standards are designed for a wide range of industries. So, unlike the pharmaceutical manufacturing sites, some other manufacturing sites could be a lot more contaminated, or a potential of contamination might be a challenge there. So these acceptance criteria has been set to cover different industries. So it's good to think about what the acceptance criteria you're setting. A USP general chapters highlighted the acceptance criteria, and I think this are quite good realistic acceptance criteria. For bacteria, it's a 3-log reduction, so 1 log less. Fungi is undefined, but you can perform a risk assessment and look at what would suit your manufacturing site. Bacteria spores, a 2-log reduction as well. So those data you get from your disinfectant manufacturer will help you assess if your disinfectant is good enough in a suspension, or if they've performed a surface test against bacteria spores. Are they effective enough? So this is a list of contact times that has been listed in the BSEN standards. 5 minutes for bacteria, 15 minutes for fungi, and for spores 60 minutes. Now you can find a lot of this sporicides that can perform better and can be effective against bacteria spores in 10 minutes. And this can change depending on the organisms, because bacteria spores can be quite a challenge depending on what the organism is. It's good to remember that you can adopt this contact time. However, if you use a 10 minutes contact time on site and your disinfectant is effective against fungi on a 10 minutes contact time, that is good. But you can go for testing a shorter contact time for bacteria, or shorter contact time for all the organisms, and just establishing that is effective on the shorter contact time. That gives you a lot of room for changes in the future. So that way you wouldn't have to requalify, but it's not acceptable to perform the test on the longer contact time than what you use in practice. Now, let's move on to a summary of the practical tests. Now, this is just I won't go into too much detail about the practical test. I've chosen the BSEN 13697 because it is a good, well-rounded test method, because it covers the worst case scenario and it does mimic what happens on a manufacturing site. So the first phase is to have a mixture of albumin solution plus the organism. Now, the organism, the starting inoculum before making this solution would be up to 10 to the 8, and the combination is dried on the surface just to mimic how a contamination will be presented in your manufacturing site. Now, it's good to highlight that this phase of the test is a critical phase, because depending on the organism, it can be challenging to keep them viable at a dry state, particularly organisms like Pseudomonas aeruginosa, sometimes even Candida albicans, can be challenging to maintain them at a viable stage throughout the test. So the next phase will be to add 100 microliter of the disinfectant on the surface of the the dried organism. And this is to mimic what you do during your disinfection process of spraying the disinfectant on your surface, on your surfaces. So this is what this stage aims to achieve. Then you leave it for the contact time. So if you have your a 10 minutes contact time on site, you leave your disinfectant for 10 minutes. And after that, straight on, you neutralize the effect of the disinfectant by adding the surface to a neutralizer. Now, the neutralizer will contain glass beads. This is used to remove the surface from remove the organisms from the surface. This is done by vigorous shaking. And once you've done that, it is plated out and left to for a number of days. Now, the log reduction is determined by comparing the recovery to a water control. Now, the same process is performed using water. Now, this is a more accurate way of estimating the log reduction. So this is another advantage of this test. And I will say, without going into much details, this test have a number of controls like that ensures that the kill you get is due to the impact or the effectiveness of the disinfectant. So you have a neutralizer toxicity control, just to make sure that the kill is not due to the addition to the neutralizer. You also have a dilution recovery control as well as a control just to look at the end of the test, the amount of organisms left on the surface. So have the organism not been recovered because the neutralization and the bead step wasn't effective enough to remove the organisms from the surface of the organism from the surface. So it's quite a robust test. Now, quick summary about the advantages of this test. It mimics the disinfection process in a manufacturing site. It covers the worst case scenario. Contact time can be adapted to process. And again, about the recovery phase, it is a more accurate way of getting the amount of log reduction your disinfectant has achieved. Now, a few disadvantages about this method is the viability phase. The drying phase can be quite challenging, but with the right level of experience, this can be achieved. And this brings us to the next disadvantage. You actually require experienced analysts to perform this test. Now, other considerations when embarking on a clean validation, always remember to qualify your environmental monitoring media. Is another key expectation to make sure that your residues of your disinfectant are not interfering with your environmental monitoring. So if you use swabs or contact plates as part of your environmental monitoring process, you want to make sure you're not recovering, getting a false negative because of the impact of residual disinfectants left on the surface. So it'll be good to qualify. It's very important to qualify this. There's a number of tests you can perform to qualify this, and make sure you have the right type of media in place with the right level of neutralizers in place. This presentation does not cover the health and safety aspect of of choosing your disinfectant, but it's good to put that into account. So this would depend on your site. Work with your health and safety head when choosing a disinfectant. Now, requalification is another key consideration. Now, I get asked this a lot around requalification. Requalification is required where there's a change in cleaning and disinfection process or change in facility. So if you move into a new facility, or example of these changes would be or new facility or formulation change, disinfectant has been changed. Change in contact time. So if your cleaning process, you've added you've changed your contact time, you've made it a lot significantly longer than what you've or shorter than what you validated, you need to qualify that. Changes in environmental monitoring trend. Are you getting a high level of environmental monitoring? Is this because your disinfectants are not effective anymore? You need to look at this and see, requalify, just establish that your disinfectants are still fit for purpose. Changes in your hold time and changes in the containers you store them in. These can have an impact on the performance of your disinfectants. So when you have these changes in place, you might have to look into requalifying your disinfectants. Just a summary, I'll round up on this presentation. I'll just look back at what we've talked about. We talked about the importance of qualifying your disinfectants. It's important to establish that your disinfectants are fit for purpose. You do not want to go by the data that's been presented by the disinfectant manufacturer as part of the bottle claim, because this has not been done under the conditions they use, so they're not fit for purpose until you perform your cleaning validation. Also talked about ensure you have the right range of disinfectants, have a bactericidal, fungicidal, and sporicidal agents. And we talked about the fact that widely used disinfectant like 70% IPA is not effective against bacterial spores. We talked about taking the necessary steps to avoid pitfalls, so you can get this right the first time. Collect as much data as you can from your manufacturer and talk to specialists. If you're using a contract testing lab, talk to them. They perform tests for the disinfectant manufacturer as well as end users. So the likelihood of them coming across your disinfectant is very high. So they can right away tell you, you know what, we have data, and we can tell you really this disinfectant is not as effective as you think it is, and they can guide you. They can you make use of those resources to make sure you save time and get it right the first time. Choose the right method. So we talked about the methods that are available and how the BSEN 13697 can be used to establish that your disinfectants are fit for purpose. And perform the study to determine whether or not disinfection process is interfering with your environmental monitoring. So if you use contact plates or swabs, perform a qualification to make sure you have the right level of neutralizer in your plates. And very importantly, you need to make sure you document this process at the end of your cleaning validation. Document this process. This is an expectation from regulators. They will expect to see a documented process. They want to see the rationale around your whole process, why and how you've used some scientific data as your backing. So thank you very much for listening. I'll be happy to take any questions. I'll pass on over to Lisa.
Thanks, Darlington, and thank you for sharing that comprehensive insight for us today. You're right, we've had a couple of questions come through as the session's been going on, so we'll take a few minutes just to look at a few of those. If we don't get to your question today, don't worry, we will follow up with you separately afterwards to get in touch with you and answer those for you. So Darlington, one of the first questions that we've had was, we have performed cleaning validation in the past using the suspension test. Do we have to requalify our disinfectants again, even though they performed beyond our set acceptance criteria?
Thank you for that question. It just I'll just draw your attention back to something we covered earlier about how the suspension test does not establish that your disinfectants are fit for purpose, because a disinfectant that might perform really well in a suspension, because the contamination is presented in a suspension, might not perform as well on a surface. So to meet the criteria, I would recommend you perform a surface test. So the data you have from your previous qualification process is good data, but I'll really recommend you look at it again and perform a surface test. I hope that answers your question.
Thanks, Darlington. Another question we've been asked is it acceptable to use a similar surface when we cannot obtain the exact surface in our facility?
Yeah, I do appreciate the fact that it can be really challenging to get the exact surface sometimes, but just to bear in mind, it does have an impact on the results you achieve. An example in the past is a site where the regulators have challenged a surface based on the fact that the surface on site had a special kind of coating, and when they came back to sourced the surface, the exact surface, and performed the test, it did not meet the acceptance criteria. So I think where, depending on how critical that surface is, I think it's very important to perform this clean validation, perform the test on the exact surface, and just work with your supplier and see if they can help you with this.
Okay, final question for today, as we're getting close to time. Darlington, we are requalifying our disinfectants every 2 years. Do we think this is necessary?
Well, I think the first question is to look at the reason behind the requalification process you have in place. It will be difficult to give you a yes or no without looking at the whole process and the rationale behind requalifying every 2 years. But as mentioned earlier, when you have a significant change in your process or your facility or your services, or I'll recommend requalification. But with the absence of that, there isn't any need for it. But look at the rationale around why you requalify every 2 years. Is it because you have a change in your process every 2 years or every year, and this is aimed at making sure that the disinfectants are still fit for purpose? So I wouldn't say no, it's not required. I would say look at the data behind it, why you're performing it, and add what we've talked about today and see if it's something that is needed or not. Hope that answers your question.
Thanks, Darlington. Thanks, everybody, for joining us today. This brings us to the end of our webinar and the end of our cleaning validation series. Hopefully you found today's session useful and collectively the webinar series useful in your cleaning validation assessments and requirements. If you've got any additional questions or you feel that you might need some additional support from us, please do get in touch with either Darlington or myself. Our contact details are on the screen, and we'd be happy to help you through those. Thanks again for joining us, and hopefully you'll join us again on other webinar series that will run in the future. Good afternoon.